Welcome to the RIMA data preprocessing video. In this video, we will cover how to run the preprocessing modules of the RIMA pipeline. These include read mapping or alignment with STAR, quality control with RCQC, gene quantification with SALMON, and batch correction with LIMA. We will cover 4 steps in this video. The first is setting our parameters and run details. The second is testing those parameters and run details. The third is running the pipeline. And last, we will check our results. Step 1, set parameters and run details. For this step, we have to check and or modify 2 files. 1, config.yaml for setting the parameters, and 2, execution.yaml to choose which parts of the pipeline we will run. Let's first take a look at the config.yaml file. The config.yaml file can be found within the RIMA main directory. When you open the config.yaml file, you'll see at the top the pointers for the metasheet as well as the reference files. For more information about these settings, please see our previous videos. For the pre-processing modules, you'll want to make sure the ref-seq-ref is set correctly. In this case, it's typically set to housekeeping for housekeeping genes. If it's not set to housekeeping, ref-seq-reference will use a downsampled set of RNA-seq files. For mate, you will set whether your files are paired end or singled end. And for batch removal and PCA analysis, you will set which covariates in your metasheet you would like to use for batch correction analysis. Next, we will modify execution.yaml to set which parts of the pipeline to run. Execution.yaml can also be found in the RIMA main directory. In execution.yaml, only preprocess individual and preprocess cohort need to be set to true. All other flags can be set to false. After setting our parameters and run details, we want to test those settings before running the RIMA pipeline. To check that your settings are correct, perform a snake-make-dry run. To perform a snake-make-dry run, use the end flag on the snake-make command. For more information about snake-make-dry runs, please see the snake-make documentation at the URL in the bottom right. When you run the snake-make-dry run, you'll have an output that should be all in green if your settings are correct. At the end of that output, you will see job counts. In this case, we ran 12 individual files, so some of the jobs will have a 12 count, and the cohort jobs will have a count of 1. If your snake-make-dry run is successful, you're ready to run the RIMA pipeline. To run the RIMA pipeline, use the basic snake-make command that you see here. There are additional options you can add to the snake-make command. To understand those options, please see the snake-make documentation at the URL on the right. The RIMA pipeline will take some time to run with the pre-processing modules. It's recommended that you run the pipeline in the background with a command such as nohub. Once the pipeline is finished running, you are ready to check your results. Results can be found in one of 2 subdirectories within the RIMA main directory. In the analysis subdirectory, there are subfiles for each of the types of modules that will run. And inside those are most of the individual raw output files as well as some of the cohort results. In the files subdirectory of RIMA, other images and reports can be found. Here you'll see some of the outputs from read mapping using STAR. Under the analysis subdirectory, in the STAR folder, you will see individual sample output that is typical for a STAR run. In the files subdirectory, you will see a cohort level report called STAR Align Report that combines all of the log outputs from each sample into one file. There are many different outputs from RCQC that will be of interest. In the analysis subdirectory, in addition to finding files for each sample, you will also see subdirectories for things such as read distribution. And in the read distribution subdirectory, you can find a cohort level report. In the file subdirectory, you will find a folder called gene body coverage. Inside there are folders for each of the individual samples with their respective gene body coverage curves, like that seen on the right. In the file subdirectory, you will also see a folder labeled TIN score, And in there is a cohort report that includes the mean and median TIN scores for all samples run in your pre-processing run. All salmon output can be found within the analysis subdirectory. In the folder called Salmon, there are individual folders for each sample, as well as cohort reports, which include salmon, TPN, and sample CSV, which includes for all samples all the TPM levels called by salmon in the transcript and BAM logs. RIMA also converts these TPMs from ensemble IDs to gene symbols, and that cohort report can also be found within the analysis subdirectory. If batch correction was performed during your pre-processing run, you will find PCA plots before and after batch correction with LIMA within the Files subdirectory. These will help you determine if batch correction is necessary for your analysis. To summarize, we have just covered the pre-processing modules of RIMA. We first set our parameters and run details by checking and modifying the config.yaml and execution.yaml files. We then tested the parameters and run details by running a dry run of SnakeMake. We then ran the pipeline and checked some of the results. Once you have run the pre-processing modules of RIMA, you are ready to move on to differential expression and immune-specific modules.